So as many of you know, the mission of my podcast is to host founders of the most disruptive companies in the world and to explain what they do to my audience in a format that's digestible. Beam Therapeutics is pioneering the use of CRISPR-based editing to develop a broad portfolio of advanced genetic medicines. Your groundbreaking base editing technology allows us to make permanent, specific edits to single bases in DNA and RNA without cutting the strands of the DNA helix. So today we'll hit on some of the revolutionary science going on at Beam Therapeutics. We'll chat with John Evans, the CEO of Beam Therapeutics, about his journey prior to his arrival at Beam and what life has been like since joining the Beam Therapeutics family. So I have so many questions I would love to ask, but for the sake of time tonight, I boiled it down to some of the most burning questions. So it's my honor to have you to join Making the Cut podcast, powered by Pounding the Table Network. And congratulations to Beam on your recent INDs as you make the next step to bringing potential curative therapeutics into the clinic. We've all been waiting patiently for that announcement to come, John.
Thank you. It's great to be here, and really appreciate this chance to talk with your audience, and appreciate everything you're doing to just try to bring more education and awareness to the space. It's really exciting.
Awesome. So John, without further ado, the first question is that I believe it was around 2017 where you met with David Liu at his office at Harvard. And base editing really captured your imagination. So at the time, you were doing some work for Arch Venture Partners. You mentioned word for word that it was clear right away that Beam was a potential rocket ship. Dr. Liu is also on record saying that at the moment he met you, he knew you were the right guy for the CEO position. So are you able to share with us some of those preliminary conversations that took place with Dr. Liu? What do you think he was looking for in a leader? And what was it that really attracted you most to Beam's base editing platform from the get-go?
Yeah, great. So I love a good origin story. So maybe I'll go way back just to start. So I was actually originally an English major in college. So I share that only to give your audience a sense that it isn't just science PhDs that can navigate this space. But it's been quite a strange trip for me. But I learned early that I love medicines, and I really wanted to work closer in therapeutics. And then also, as I got into the pharma industry, I started to realize that I wanted to work in smaller and smaller companies, because that was just really where you could see it all come together, and the moving faster and the innovation level was going up, right? So the most recent company before Beam is probably worth a comment, because it gives you a good sense of why Beam resonated so much with me. So that was Agios. So Agios is a small molecule cancer company that targeted metabolism, is looking after cellular enzymes and metabolism, and trying to treat cancer with that. And amazing company. And we ended up focusing on genetic mutations in some of these enzymes. And we had drugs that literally would turn off those mutations. So you're basically fixing what's wrong. Mm-hmm. And we call that precision medicine, because what that means is you basically know who to treat when you go to the clinic. So you're not spending phase 1, phase 2, phase 3 searching for signal and then trying to statistically show that your drug has some activity. You're right away treating patients who have the mutation, and your drug should fix it. And so sure enough, very early in phase 1, we saw activity. We saw complete responses in patients with acute leukemia who should be dead in a couple of months otherwise, right? And what that means is that you now really know you have something. And so now you can work with the FDA and with patient groups to move as fast as possible. And so we ended up getting 2 drugs approved with this sort of similar mechanism in less than 4 years from IND. Now that's like light speed, but I think in general, these precision medicine approaches are always going to be faster than the 8 to 10 years that it normally takes. And you just have higher confidence in what you're doing biologically. So for me, that was really powerful. The frustration at Agios was that in small molecule world, if you do it once, it's great, lightning strikes, but then to do it again, you're basically starting from scratch. New targets, new chemical scaffolds, and it's just the whole journey and all the same risks. So that was frustrating. Agios is going to get there. They're going to get some new drugs soon. But so I left Agios. It was the right time. And so I got involved in Beam. And so as you said, I met David Liu, and I'd never met him before. And it was an incredible experience. And he talked me through base editing. And I did not know a lot about CRISPR at the time. And I certainly didn't know about the nuances of Gen 1 nucleases and scissors versus what base editors can do. And I know we'll talk about all of that. But what really struck me was 2 things. One was that this was sort of the ultimate precision medicine, right? Because now every patient we could treat with this system, we will know who they are based on their genetics. And then we'll have a drug that can potentially reverse those genetics back to normal. So just like my Atlas experience, these were going to be clinical paths where we really know who to treat, and we really think it should work, and it should work early, and it should set up a fast path to getting an approval and getting it to patients. But even better, it's a platform.
Right.
Because these are basically programmable. And as I know your audience knows, the beautiful thing about CRISPR and the reason why it was such a breakthrough over zinc fingers and tals and other things that come before it is that easy programmability. The guide RNA encodes the address in the genome where we want to search and then bind, change that little element, and you have an entirely new medicine. And yet everything else about the medicine could be exactly the same, the manufacturing, the regulatory package, all of that. And so that meant that with base editing, if we could do it even once, we could do it 10 or 20 times relatively quickly. And so that really was what captured my imagination. So I couldn't sleep that night. I was sort of just thinking constantly about what was possible here. And I think the elegance too of being able to think about single letter changes within the code of DNA and using that to basically reprogram the outcome of what the gene is coding for, whether you're fixing it or just altering it or modifying it functionally, the possibilities are really endless. And that's certainly where I think David and I did hit it off very early. I think sharing a common love of science and a common sense of urgency, I think, to get these technologies to patients as quickly as we can, and a little bit of shared just geeking out on what was possible with the technology. And we went off to the races. And of course, we were joined by Feng Zhang from MIT in the Broad, and then Keith Jung from MGH, just incredible scientists in the CRISPR field. And that was sort of the original group that helped set up the company.
And so, John, bringing novel medicines into the clinic comes with a great deal of responsibility. While Beam does away with the double-strand break, it's certainly not free of other precision chemistry risk factors. So bystander editing is editing of one or more non-target bases in addition to the target base in the activity window of a base editor. And translocations are another risk factor, which leads to chromosomal abnormality due to the double-strand breaks. So at Beam, how do you think about these risk factors? What have you seen from preliminary animal models, assay tests, and how far do you think we've come in our abilities to study cell changes at the single base resolution?
Yeah, great question. So maybe to answer this question, let me do now the mini PhD for your audience in terms of what base editing is. And I know a lot of people already know this, but it's probably worth doing.
Right.
So, when I think about all these editing tools, I think of them actually in the context of the general push for gene therapy, which is we want to have one-time curative treatments. And with gene therapy like AAVs or lentivirus, you're basically adding some extra gene into the cell. You can't control where it goes, but it'll just go somewhere, but hopefully it expresses and that may help. The idea has always been it would be better to do gene editing. Why don't we just work with the genes that are already there and hopefully fix them or correct them? And so the first generation of gene editing tools had one really important breakthrough. And this was, again, zinc fingers and then talons and then famously CRISPR. And that was how you target. So if you think about it, every one of your cells, A, G, C, and T, has 3 billion letters in it, and you have to find a single address out of those 3 billion. I mean, that is an incredible challenge. And those different tools can do that. And it's really quite remarkable how they do it. Obviously, CRISPR, the ease of it and the flexibility is why it's so popular, but all of them have that ability. So that's amazing. The drawback in Gen 1 is once you get there, the only thing you can do is cut. Okay, so you make a double-stranded break, which is like a cleavage. And then really at that point, your job is done and you're leaving up to the cell what the cell will do next. And the cell basically doesn't like that. It's a genotoxic event. So it'll put the pieces back together again as quickly as it can, but it does so with damage. So you get random insertions or deletions of letters at that target site, basically scrambling the sequence at random. And so that will basically disrupt a gene, and it's good for knocking things out. But obviously the randomness of that and the lack of control isn't so easy to control the outcome of the edits. You don't have that precision control. And so that was really Gen 1. And obviously that's moved forward, and now there are places where you can knock something out and have some therapeutic benefit, and those are the programs that we see now moving forward. Beam really comes out of Gen 2. And so it was some scientists in David Liu's lab, including Alexis Comer, Nicole Gaudelli, who said, isn't there a better way to do this? And so what we came up with was base editing. And so what base editing does is it uses CRISPR for that same targeting ability we love so much. But once we get to the target site, we open the DNA up and we use a deaminase, which is a naturally occurring chemical enzyme, to edit one letter to another. Okay, so we make this very precise single letter change. We don't need to make the double-stranded break. So that's the critical answer. So now in thinking about your question about sort of what are the consequences of our editing, what are the kinds of off targets we think about. So the first important breakthrough is that we don't make the double-stranded break, and that's really critical. And so double-stranded breaks have lots of consequences in cells, and the more of them you make, the worse it gets. So there are damage pathways that turn on, the cell viability can get worse, and particularly you can end up with chromosomal changes, because if you think about it, the cell doesn't always know which pieces go where. And so sometimes it can make that mistake. And so if you make a mistake, you'll get complex, novel chromosomal aberrations, and that can be quite concerning. So generally, I am of the opinion that moving away from double-stranded break editing whenever possible is going to be preferable. And so that's one thing that base editing does. Now, you noted one of the features of the way base editing works is there is something called bystander editing occasionally. And so the way that works is basically what base editing is doing is it's actually opening up a little bubble in the DNA. And it's a short window of about 4 to 5 bases wide. And the deaminase we use will recognize only one kind of base. So if we use an adenosine deaminase, it will only recognize an A. Cytidine deaminase will only recognize a C. And so if there are more than one A or C in that window, you can potentially edit multiple bases. And there's no particular way to stop that other than to shift the window or re-engineer your editor, which we actually can do. So our job is to target the base that we want to target in the middle of that window and then see if there's any bystander edit that we have to be concerned about. The bottom line is bystander editing really has not slowed down any of our programs and is not something that we're particularly concerned about. And if you think about it in a couple of ways, I mean, if we're trying to sort of repair a point mutation that's causing a disease, let's say, so you have a broken gene and you want to fix something that's wrong, okay? So let's say we make that edit successfully. If we edit something else, then you have to see, does it have any consequence? And actually, every third letter we edit has no consequence because of the way that the amino acid code is constructed. And so those are just synonymous changes. And then if there are other edits, they may be at very low levels, and most of them are inconsequential to the function of the gene. You've still restored the function of the gene. In the worst case scenario, if it was going to disrupt the function of that gene, you've just done a round trip. You fixed it and then you broke it again. so there's not going to be like a toxicity question or a safety question. You just failed to cure that cell. And so, these are things that you can basically test very quickly in vitro and identify whether that's something to be concerned about. And if so, you can engineer it and work on it. The thing to stress is to compare that situation to the nuclease again. Right, so we have a very well-defined and precise editing profile, which is generally either just the base we care about, or if we have one of these bystanders, we have to think about the bystander. But we're still getting the same pure product every time. The nuclease, every single cell is getting a different outcome. You're getting tens or hundreds of different unique alleles based on the random outcome of that indel process. And so we kind of report indels in aggregate. We say we got 80% indels, but the reality is indels is code for a wide rainbow of different alleles, right? So the diversity of outcomes you're getting with nucleases is much greater than with the base editor. And so we really like the purity and the predictability that you get with the base editor. In terms of your question about off-target overall profiling, I think the field has actually come a long way. I think that we have great assays now to measure off-targets, both the nucleases and base editors. So I think we're getting, if you think about it, we have almost digital resolution here. There's only 3 billion things to check. It's a gigabyte hard drive, and you just do the whole genome sequencing. You see if there's been any impact, then you can understand that with total resolution. And so for base editing, we've had to come up with a few extra assays that characterize single base changes compared to double-stranded breaks, and we've done that. And now we've been through the FDA with a complete IND, and they thought our package looked really good. So I think we're in very good shape there, and I would say I'm very confident and bullish about the sort of general off-target profile of the system.
John, I'm sure there are probably so many names behind the scenes that don't quite get the recognition they deserve. So Anzalone, Gadelli, Komor are some really superstar names. They're superstar researchers. The headline being scientific achievements that most people are probably already aware of. Where do you give credit for being able to recruit top talent? And are there others that you'd like to mention today that deserve more recognition for pushing the boundaries of creative engineering at Beam?
Yeah, it's a great point. There's no question we've had some incredible science that has driven this, and science comes from people. These are just amazing minds. And so, yeah, so Alexis and Nicole, I think I mentioned before, were the pioneers who really invented base editing along with David Liu. Alexis is now a professor on the West Coast. Nicole actually followed the technology to Beam. And so she works at Beam as our head of gene editing platform technologies, and is an amazing leader. And in fact, I'd say of our first maybe 12 employees, probably 8 of them were from our founders' labs. And these are people who had seen the technology emerge and actually had worked on other technologies. So we have people who were at the early days or on the original papers of Cas9 itself, of CPF1, of many other sorts of innovations that came around at this time. So whether it be David's lab or Feng's lab or Keith's lab, we got some amazing talent in those early days. From there, you have to build a company, and we've really been growing, ever since. How do you attract the talent? I do think that the technology is so exciting. People who knew the field sort of knew this was a big deal and a potentially big breakthrough. I think the appreciation for working around double-stranded breaks, is taking more time to build up, but there were some people who said, yes, this is absolutely key. And then just the precision of the editing outcome. And then we just worked hard to create a culture where it was exciting for people to come and work. I mean, with some of these scientists, we're literally competing, not even with other companies, we're competing with that academic post where they can go be a professor or they can work at Beam. And so you think about how, you have to really create an exciting science environment to make that make sense career-wise. And I think we've done that. So I'd say early days, we had, Susan O'Connor joined us as our head of HR. She had built a lot of companies with Third Rock, and she knew how to do that and set up the right culture. So she and I worked closely together on that. And then Pino Charamella, our president and chief scientific officer, was also another early employee. And he really brought that knowledge of how you build this into a platform technology. And he came from Moderna. And in fact, at Moderna, he set up all of the vaccines division, and is even on some of the patents of the stuff that became the bedrock for the COVID vaccine, which is pretty exciting. And so I often joke that Beam is sort of a mix of some of the stuff that I bring from Agios, and then maybe some Third Rock in there, and then some Moderna from the platform. Hopefully pulling the best from all of those.
Really one Voltron.
Yes, exactly. That's the idea. But you can't predict how these things work out. I mean, a startup is a family, and it grows. And every single person who comes into the company adds something to that mix, and that's great. And it just gets richer over time.
So you mentioned the idea of culture, and then also the idea of platform. So I really love that you brought those two up, and I plan to hit on those in a little bit. So as retail investors investing in early stage biotech companies, revenues don't always hold as much weight in comparison to established companies with approved drugs. So we have to objectively grade companies based on a variety of factors. And one of the most important factors is culture, as you mentioned. So although it's a relatively small sample size that you can see online, it's worthy of noting because it's clear you've built a one-of-a-kind culture at Beam Therapeutics, and it's uncommon to see 100% approval of a CEO, especially in comparison to gene editing peers in the space. So I want to briefly read off some of the employee reviews for you here. I think the audience will actually find this quite humorous when they hear some of the pros and cons noted by the employees, because in my eyes, the cons, most definitely could be seen as pros. So we're going to start off with the pros. Amazing research freedom, incredibly supportive culture, even for personal life and education goals. Great benefits and company parties and events, brilliant coworkers and collaborative atmosphere. The best science. Incredibly understanding of time off and work-life balance. Dedication to community and diversity, gives credit where credit is due in publications and patents, which that I totally agree with, because we see that time and time again. Management is excellent, with a CEO that actually has technical expertise, who makes it a point to know everyone and who contributes intellectually to science. Great culture, good benefit, good science, honest work, diversity is celebrated, never been talked down to or shamed. And so those are some of the pros that I've picked out and I wanted the audience to hear about. Now here are the cons. Sometimes conversations are too nice. Healthy and collaborative conflict builds better systems. Rapidly growing, so we'll have to see if they are able to continue the awesome startup culture. So John, can you tell us how important it is building not only best-in-class editors, but sustainable best-in-class culture where employees are able to show up every day and put in their best work?
Love it. I just feel like I got a review. That was great. I think it's such a great question, and it's so important, and we don't talk about it enough, frankly, as an industry. So there is nothing more important than culture. It's easy to say, but if you've ever been in a company that has a great one, or if you've been in a company that has a struggling culture, you know how important it is. I do think culture is very much of I think you pick it up from an apprentice model. I think, so it's just sort of seeing how it's done right. And so again, I think, I brought a lot from Agios. I think, others of our leadership team have brought things from their past, and we've just tried to put it together. One of the things that has typified the Beam culture from the beginning is it's very bottoms up. So our value statements, it wasn't me writing them down in the corner. It was actually, we worked together for 6 or 8 months. Then we said, okay, feels like there's something happening here that's good. Let's go off site and spend an entire day and a half and try to now articulate, what does it feel like to work at Beam? And that was actually the team that came up with these various sort of vision statement and values that we have. And I think that's been a way we've done almost everything. So we have so much grassroots, energy at the company. And we want to encourage that, right? I mean, because, we knew we were going to be growing and bigger. And so if it's going to be a kind of place where it's going to be top down, where I'm telling people what to do every day, not only is that not fun and people don't get excited to work there, it's actually not efficient. Like, we knew from the beginning, the scale of what we were going to try to build was going to be really large. So it's got to be team driven and bottoms up, where people are taking accountability for the culture and for working together well and for living the values. So that's really critical. We do a lot of fun stuff too. I mean, so one of our values is fearless innovation. And so we try to really talk about risk taking and the fact that we're going to have to try things that may not have ever been done before. We may not have the answer on how we're even going to do it. And so, but we've got to try it, because that's where we're going to get these big breakthroughs. And so one exercise we did early days was we watched a lot of rocket launches. And so we watched all of the SpaceX videos where, they're trying to land the rocket and it's just blowing up constantly for 3 or 4 years. I mean, I can't even imagine how they stuck with it. And then finally they barely land one, and then they really landed, and then they land 2, and then they're launching a car on Mars, right? And so the lesson for me in that is that, 1, you have to be fearless. But 2, that once you're in these engineering loops, and I think gene editing is in an engineering loop where you're basically, hypothesize it, make it, test it, refine it, and iterate. These steady improvements, they become exponential in how they add up. And I really think that's happening with gene editing. I mean, look at Beam, right? We are an improvement on first-gen technology that was only first published 9 years ago, and we're already reaching the clinic with a next-gen version, and it's only going to continue, right? So we try to really capture that spirit at Beam, and above all else, we try to make it a real team environment where we're collaborative, we're transparent with each other, and where we're all aligned around this mission that we talk about of creating lifelong cures for patients suffering from serious diseases. That, at the end of the day, is why we do this. And if we get stuck, the science isn't working, or if we're having conflict, or we don't know the path forward, coming back to that mission is going to be the thing that gets us back on track.
John, I love that you hit on that idea of like little iterations over time, incremental iterations over time, become, larger and larger changes that, ultimately, create more growth in a company. So the next topic I want to hit on is that you recently acquired the lipid nanoparticle discovery platform, Guide Therapeutics, for $120 million upfront and up to $320 million in milestone payments. So Guide was founded in 2018 and uses DNA barcodes to test hundreds of LNPs at a time for their ability to deliver genes to different cells and tissues. So during ASH, over the weekend, Beam, I believe, presented updated preclinical data using this technology to screen more than 1,000 LNPs for potential to deliver to HSCs, and identified LNP HSC1 as the most potent. So with efficient transfection in both mice and non-human primates. So there's a lot to unpack with this. So I'd like to approach it in several parts. Could you explain the DNA barcode strategy? What exactly is that? And how are you able to screen so many different lipid nanoparticles for delivery to different organ or tissue types?
Yeah, great. So we've talked a lot about editing. Editing is critical. We think base editing is a potential best-in-class technology, and everything about our strategy is designed to get it as broadly as we can, as fast as we can. So we famously are doing all of the different delivery modalities and therapeutic areas that are currently accessible in genetic medicine, we're going to go there because we think base editing has something to offer. So that leads us to delivery. And so in genetic medicine, delivery really matters. These are not pills. You can't just take a pill and have it go to the right cell. This is complicated machinery. You've got to get it in carefully. So we have a variety of ways to do that. One of our favorites is the lipid nanoparticle containing mRNA. And so what we put into it is that the mRNA encodes for our editor, so that once you're in the right cell, it'll express the editor, that's the protein. And then you also put in the guide RNA, that short little RNA element that's the targeting. And so those come together, goes to the nucleus, searches, finds, edits, and then washes out of the system, and you're done. So that's a very complicated process that we need to trigger. So much like the COVID vaccines, we make lipid nanoparticles with mRNA in them, and that initially is going to go for our liver programs. And so if you put that into the bloodstream, it'll go to the liver, and we've optimized it so that we're getting very good liver penetration now, and we can indeed edit the liver with high efficiency, which is great, at least in primates so far. But LNPs are so good, we'd like to see them do more, right? You know, the great thing with LNPs is they're synthetic to manufacture, they're highly scalable. You can redose them, so you can, you know, add a little more, you're going to get a little more editing. They have a larger packaging capacity than some viruses. There's no pre-existing immunity that you have to worry about like some viruses, and ultimately lower cost of goods, which is, you know, if we're going to treat everyone around the world, which we would like to, that's going to be important over the long term. So we've been thinking about, could LNPs go to more places than just the liver? Now, the liver is the big, you know, sucking everything out of your blood. So that's an easy target. Everything else is going to be harder. But this company, you know, Guide and this team that had done some really pioneering work to create a system to try to screen LNPs to go to other places. And so basically what they did is we add DNA barcodes into that system that are unique. And you basically make a whole bunch of different recipes for your LNP, different cationic lipids, different formulations, different process. All of that matters, actually. And so once you do that, you have tens or even hundreds of different lipid nanoparticles. You can then basically put all of them into the animal at the same time. And this is the really clever part. And so you're basically doing massive high throughput screening. I mean, that would be a single experiment every one you did otherwise. And then you basically look in your tissue of interest, right? So in our case, we might go to the blood stem cell and say, which of those LNPs got to this cell? And then you just read the barcodes out and it tells you, okay, these were the recipes that got them. And so, and then the even better part is you're screening the whole body every time. So it's not like I have to choose the blood cell. I can also check the muscle and I can check the brain, and we're going to learn that information, and you might get different answers, of course, right? Different LNPs may be good for different organs. So we are very excited about that. So the first lead formulation coming out of that work is going to be targeted at the blood. That's of course of great interest to us given our lead programs in sickle cell disease. So we're very excited about that. And so yeah, at ASH we showed we showed a little bit of mouse data early in the year. This time we showed expression of mRNA payload in up to 20% of the blood stem cells of a non-human primate at a clinical, you know, relevant dose. And that is at or right at the front of the field, I think, in terms of people thinking about how to do this. And so we're very excited about that. That's obviously the subject of a lot more lead optimization to come. But you know, ultimately, our long-term goal, our sickle cell programs will begin treating patients by editing the cells outside of the body and using a transplant to then put the cells back in. Long-term, we would love to switch that over to an in vivo edit where all you do is you put it into the bloodstream, goes to their marrow, edits, and they're done. And of course, there's many more things in hematology to do with that kind of technology beyond just sickle. And so it's obviously, you know, very interesting. From there, the Guide platform, we are screening many other tissues, of course, that are of interest, whether it be T cells or CNS or muscle or heart, lung. I mean, these are all of interest, and you know, we'll have to follow the science and see where we get the most traction and what that opens up therapeutically. It's not our only bet in delivery. We're actually doing a lot of different things in the delivery sector, but that's a big one, and we're quite excited about it.
So I'd like